Molte grazie.
Thank you very much for inviting me to participate in this interesting meeting. I'm learning new, very inspiring things. Well, a few technical problems with my presentation. Now, I will now present our 10-year work that has been supported by multiple bodies, including Mariani Foundation in Milan, Emilia-Romagna region, and the Ministry of Health. The Ministry of Health gave us a significant amount of funds to support our activity. Now, I will be presenting a few data, and I hope they will be convincing. They will sound convincing. Neurosteroids and allopregnanolone, in particular, are endogenous modulators of seizures. So apart from its pharmacological effects, it is also recognized from the physiopathological point of view. Now, very rapidly, let me share a few data with you. Of course, this is based on an experimental approach. Neurosteroids were first identified many years ago at the beginning of the 80s of last century when they demonstrated that orchiectomy and adrenalectomy, i.e. the removal of tissues that can synthesize steroid hormones, would leave in the brain the concentration of DHEAS and PS unchanged. These data were published by the Biagini group. So they demonstrated that the brain could synthesize these two steroids regardless of the endocrine axis. And then they found that the most important neurosteroid is allopregnanolone, which is synthesized in many regions of the central nervous system, while DHEAS is present in high concentration, especially in the hippocampus. In both cases, the common precursor is pregnanolone, which in the brain is directly synthesized by cholesterol thanks to the activity of the P450 enzyme SCC. This is a mitochondrial enzyme, and it can also be obtained thanks to the inversion of pregnanolone sulfate, i.e. the peripheral source of this precursor steroid, through the activity of an enzyme that is called sulfatase. Pregnanolone is then converted into progesterone, and the progesterone is the common metabolic intermediary for both pregnanolone and another neurosteroid, which is very much studied for its epileptic activity, i.e. tetrahydrodeoxycorticosterone. However, the same enzyme of the periphery was not found in the brain tissue. Now, why are these steroids important? First and foremost, because they link to an inhibitory receptor which blocks seizures, GABAA, and allopregnanolone, as demonstrated by Maria Majewska, it links with it as an agonist, while a pregnanolone sulfate links as an antagonist. Allopregnanolone, the most important antagonist, has an effect on GABAergic transmission by strengthening it. It also has an impact on phasic phase currents. It slows out the current decay, and it also strengthens the inhibitory effect of GABAA, as demonstrated in the case of the study of current, the so-called tonic current of the GABAA receptor. This current seems to be of particular importance to combat seizures in the central nervous system. Now, additional evidence, and this was found at the end of the 90s, Rogawski's group demonstrated that the anticonvulsive properties of progesterone, which were known already in the 40s, depend on its conversion to allopregnanolone. This evidence was found by blocking the synthesis enzyme, i.e. 5-alpha reductase and 5-alpha hydroxyprogesterone, which is a precursor of allopregnanolone. By administering this drug, they could block the progesterone effects. Let's now have a look at the evidence in favor of the the role played by this endogenous synthesis mechanism, i.e. allopregnanolone, and modulation of seizures, which originate in the brain tissue. Our model, the model that we studied, was the model of the temporal lobe. Very often it is a pharmacoresistance, and it is often associated with a lesion in the hippocampus. This is a structure which plays a role in this type of epilepsy, and it is known as hippocampal sclerosis. Neurons get lost and glial cells proliferate. So we injected pilocarpine to induce seizures. These seizures lead to status epilepticus, and as it can be demonstrated through the typical EEG tracing, this is what we obtained by injecting pilocarpine. So for 48 hours, we might have some critical episodes, generalized convulsive seizures, up to for a period up to 48 hours from the administration of pilocarpine. And then on day 3, there is a recovery, and you will no longer notice any epileptic activity, with the only exception of new episodes in the next days. And this is what you can see through the EEG. So if you look at the animal, if you observe the animal and register its motor activity, you will not notice any evident seizures. This till day 14. The electrical activity precedes the motor activity. Witness this video, and the video shows the so-called stage 4 seizure using a Crescent scale. This scale classifies seizures according to their severity, and the maximum is 5. This is the highest value. And we observed the onset of seizures in rats treated with pilocarpine, and of course we recorded these seizures according to the metabolic activity leading to the synthesis of allopregnanolone. Now, this type of study has allowed us to characterize a relation between the increase in the metabolic activity of the enzyme that limits the synthesis of pregnanolone, P450 in relation to the onset of seizures. Now, after the administration of pilocarpine, this enzyme is induced, i.e., it is synthesized both in the neuronal cells that are here shown colored with a immunohistochemical reaction in the hippocampus in particular, and in the C3 area in particular. It is not very present in the glial cells. It is induced in the neuronal cells and in the glial cells, which are here enlarged. They are shown enlarged with an enlarged view in this panel. And it is in the glial cells that we observe the phenomenon. We quantified this phenomenon and noticed that the phenomenon leads to a long-lasting expression of the enzyme, which reaches its highest peak 3 days after the induction of the epileptic status. It is maintained for 7 days, and it starts to decrease after 2 weeks, and eventually it is still at a very high level 20 days later, although, of course, the level is smaller compared to the peak. Now, in the neurons, we notice a transition, especially during the first week after the status epilepticus. We tried to identify these cells which would stain for P450 after epilepticus status in an area that was not populated by neurons. And we noticed that they are mainly astrocytes, glial cells, which are activated. They show P450, although immunoreactivity was also found in microglial and oligodendrocyte cells. And here is another important feature to be mentioned. These phenomena were limited to the hippocampus. In nearby areas, for example, the cortex, there were smaller variations and non-meaningful variations, especially in the entorhinal cortex. And the most interesting thing or at least for me and my team, was to demonstrate that the induction of the enzyme was proportionate to the duration of the status epilepticus. Status epilepticus can be interrupted by administering anti-seizure medications. So by stopping it at an early stage, you will notice a reduction in the induction of the enzyme. So the longer the status epilepticus and the peak was observed after 3 hours, the more the enzyme is induced. Now, we observed the onset of the seizures, stage 4 and 5 in our animal models, and we noticed that in the rats where the status epilepticus was longer, we also had a delayed and incomplete onset of seizures, stage 4 or 5 of Rasinski's scale. So with a greater induction of the enzyme, you would have a later onset of the seizures and in a smaller number of animals compared to those in which the status epilepticus was shorter. Moreover, in this group, the group that underwent a status epilepticus for at least 3 hours, there was a close correlation between immunoreactivity and the onset of seizures. So as soon as immunoreactivity starts to decrease, we notice the onset of the seizures, and the number of animals in which we have an onset of seizures progressively increases as immunoreactivity decreases for P450. So our conclusion was that there is evidence in favor of the fact that P450 SCC is positively associated with delayed onset of seizures. We then characterized the same animal model, but in younger animals. This model is normally started on 8-week-old rats. And we tried to understand what happened in 3-week-old rats. And we knew from literature that in these rats it is more difficult to have stage 4 and 5 seizures after the induction of the status epilepticus. We confirmed the data in literature in young animals. 1 month away, only 20% showed seizures, stage 4 and 5 of our scale. And in these animals, we observed that the induction of immunoreactivity for P450 SCC was remarkably higher compared to adult rats, in which stage 4 and 5 seizures appeared in all cases. We then concluded that the induction of P450 is associated with less severe seizures after the appearance of spontaneous seizures following a status epilepticus. And then we wondered, following the increase in the enzyme that is so important for the synthesis of allopregnanolone, so is this followed by an increase in the concentration of allopregnanolone in the brain tissue? We mainly studied the concentration of this neurosteroid in the hippocampus using a technique that we tend to prefer for this kind of array, i.e. liquid chromatography associated with mass spectrometry. And we noticed that in rats treated with pilocarpine, we had a higher concentration of allopregnanolone compared to the control rats. And not only that, in addition to that, this difference could also be eliminated by administering, as Michael Ogalski's group did for normal rats, by administering finasteride, i.e. the drug that can block the conversion of progesterone into a precursor of pregnanolone. Moreover, we also studied the potential consequences of this increase in allopregnanolone, and we focused on the electrical activity of the neurons in particular, and this is what we characterized. We characterized the tonic inhibitor current of GABAA receptor. Usually it is measured in voltage clamps, and we had we added a GABA receptor antagonist in our solution, and what we demonstrated was that in rats treated with a pilocarpine, GABAA tonic current was higher in an area of the hippocampus that's called the subiculum. So there is a P450 SCC induction that is associated with a seizure reduction. There is an increase in the synthesis of allopregnanolone and also an increase in the GABAA tonic current, which is accountable for the delay in the appearance of the seizures and also accountable for the fact that they are less severe in the young rat. And then, in order to confirm our hypothesis, we treated our animal models with finasteride. So look at the observation in vivo on our animal models. The experiment was carried out on adult rats first. They had been in a status epilepticus for at least 3 hours, and we noticed that finasteride could remarkably anticipate the onset of seizures during the phase following the induction of status epilepticus, i.e. The latent phase, in which you commonly don't have stage 4 or 5 seizures of Racine scale. So finasteride worsened the situation in these animal models treated with pilocarpine, which, of course, leads to spontaneous seizures. This was not the case in the rats that had been exposed to short status epilepticus, so the induction of P450 had not been meaningful in these animals. And again, even in the young rats, finasteride anticipated the moment, the appearance of the seizures, although this was not the case for all the animals. This was the group in which we had the highest expression of the enzyme. So the conclusion was that allopregnanolone can actually control the epileptic activity during the latent period. And let's now move on to the clinical studies, which are more recent and also more incomplete. They are still lacking some data. We collaborated with the neurological department of the Modena Hospital and Dr. Meletti, and the same model of finasteride in animal models was validated in a patient suffering from epilepsy of the temporal lobe. The epilepsy was under control. It did not need any pharmaceutical treatment. However, then we decided to resort to medications due to a worsening in her clinical situation. So the patient was going through a polytherapeutical approach, and we found out that she was being treated with finasteride for aesthetic problems because she had much hair on her body. So the pharmacoresistant seizures no longer occurred, and this confirmed our hypothesis, i.e. allopregnanolone has a major role to play in patients suffering from epilepsy, so not only in the animal model, but also in the latent phase. And then, thanks to the collaboration with our research center, we carried out experiments to monitor the level of allopregnanolone, also in patients suffering from PCDH19 disease. We noticed and confirmed the data published by Giuseppe in June of last year, i.e. these patients show a remarkable reduction in the levels of allopregnanolone as confirmed by the difference in these two peaks, the peak in the patient versus the peak in the controls. Not only that, in addition to that, since we were also interested in the levels of pregnanolone sulfate, we dosed this steroid, which is a precursor, a potential precursor, peripheral precursor of allopregnanolone. In this case, we even observed a further reduction, thus confirming what Joseph had assumed, i.e. that as a matter of fact, mutations in protocaterin have an impact on the activity of the endocrine axis. Therefore, this confirms our hypothesis, our initial hypothesis, i.e. allopregnanolone is important from the physiopathological point of view in keeping seizures under control and preventing uncontrolled seizures when no pharmacological treatment is being used. And I would like to thank the members of staff who participated in this project, Chiara Lucchi and Cecilia Rustichelli, for their studies using liquid gas chromatography and also on the levels of pregnanolone and allopregnanolone, Enrica Baldelli and Jonathan Vinet for their studies on rat tissues, Gabriella Pannucci, who carried out the laboratory studies on tonic current of the GABAA receptor. And thank you very much for your attention.